Thank you so much for inviting me. It's really nice to be here. And yes, I'm going to give this talk with a slightly cryptic title. So we're going to see some good data, we're going to see some bad data, and we're going to see some ugly data. So as Anna said, what I really love is fluorescence microscopy. That's my thing. And fluorescence microscopy can give you some really beautiful images. For example, here's some really nice data. We've got some mitochondria in fish and yeast. We've got some actin in mammalian cells. And we've got some nuclei in mammalian cells. And one of the great things about fluorescence microscopy isn't that just that you can get pretty pictures, but you can extract quantitative information from those pictures. So you can do tasks such as segmentation, structure analysis, classification. And if you've got nice, good data, you do your analysis, then you'll only get good results. So you can see my mitochondria are nicely segmented. This middle picture is a local alignment analysis to see what direction filaments are pointing in. And the last one is a classification, so just asking what's in the image, nuclei. Now that's great, but if anyone here who's ever used a microscope knows that one does not simply just get good data. A lot of the time, our data looks a lot more like this. It looks like bad data. And now, of course, if we try and do our analysis on bad data, then unsurprisingly, we get pretty bad results. And this is not something that's limited to fluorescence microscopy. This is a kind of the universal paradigm of, to put it politely, garbage in, garbage out. So why don't we just acquire great data in fluorescence microscopy all the time? Well, it turns out that the imaging conditions that we need to acquire that good data, especially for live cells, make your sample pretty sad. It can kill it, it can bleach it, it's bad times. Whereas it turns out samples really love the imaging conditions that you'd have to collect this bad data. And that's this core problem in fluorescence microscopy. So what you're trying to do is you're always trying to balance how happy your sample is with the kind of information you can get out of your images. And we often refer to it as this kind of triangle of despair, pyramid of frustration, some kind of geometry of sadness in general. And so if you have a look at those corners, the closer you get to one of those corners, so for example, the higher temporal resolution you image at, the further away from the other corners you get, the more you're compromising other things. And to show you that this isn't just magic geometry, I have a very sad video to show you. So this is a video of its same cells in two channels. The left is the fluorescence channel, we've got tubulin GFP, and the right is just the transmitted light. And what I'm going to show you is all I'm going to do in this movie is I'm going to image these live cells, and I'm just going to turn up the laser intensity, and we'll see how they feel about it. So here we go, turning up the laser intensity. Things are fine to begin with, but then, if we look at the transmitted light channel, you'll see the mitotic cells are blebbing, that bottom cell is really retracting, and this has happened within 12 minutes. I mean, I've been incredibly violent here, unnecessarily violent, but too much light really can kill your cells. So you might think that the question that we need to answer for live cell fluorescence microscopy is not to play the movie again, because it's very cruel, but what's the best image I can acquire without damaging the sample? That's one question that we might want to ask ourselves. Now, to answer this question, what we need to do is we can actually need to know when we're damaging the sample. What does sample damage look like? How can we tell if it's happened? So there have been a few papers that have looked at this. So a few years ago, actually 10 years ago, my gosh, there was a paper from Marcus Sauer's group, which was looking at how cells survived after super resolution imaging doses. So you imaged them, took them away for 24 hours, and then saw what happened to them downstream, whether they kind of were happy or they died or they went into this weird frozen state. There was an archive paper about 8 years ago called DeadNet, what a lovely name, where they trained a neural network to try and predict from transmitted light images if cells were going to die or not. So the idea was you had a monitor where you could see early, just from transmitted light, if your sample would die. Didn't really go anywhere. I tried to get hold of the algorithm once, and it turned out the author had left and gone to IBM. So that was a shame. And more recently, Ricardo's group, my old group, have published bioRxiv, which is called the PhotoFit framework. And this is a kind of assay. It's a protocol for determining how much light your sample can withstand before it dies, using parameters like mitotic delay and arrest to figure that out. But this feels very complicated to me, and I am not a complicated person. I'm a very simple person. And so, for example, if we want to try and calibrate our imaging to our sample, we need to do that for every different sample we have, for every different cell line, for every different genetic modification, perturbation, because all of these can change our sample health. And so for me, this is too difficult. This is a great way to approach it, but for my small mind, too difficult. So I'm not interested in this question. I'm interested in a slightly different question, which is just, what's the worst looking image that I can still analyze accurately? Or to put it bluntly, what's the worst image I can get away with? Because that's as good as you can get in fluorescence microscopy. So if I want to answer that question, then we need to answer this question, and that's, how do we know if an image is good or not? So I'm afraid there's some audience participation, which is compulsory. So this is an image that I took. It's on a spinning disc confocal microscope, and my sample is fish and yeast, and the nucleus is expressing DFP. Hands up if you think that's a good image. Thank you, the one person at the back who quite confidently went up. Thank you, the few people in the middle. The rest of you quite rude. I tried so hard with my beautiful data. But what I've done here is I've asked you a very stupid question. Is this a good image? What kind of question is that? So we're going to do it again, but with a more specific question. So this time I'm going to ask you, when could you accurately measure nucleus morphology? So there's a nice question that is kind of very tangible. I'm going to turn up the laser dose on my sample, and you're just going to stick your hand up when you think you can answer that question. All right, let's go to the microscope. Some hands going up, a few more hands going up, Arne is up. All right, so by now we're all pretty confident we could have a decent crack at measuring nucleus morphology. But what's interesting is there wasn't a particular image where we all suddenly went, yeah, it's good. We all had a different idea of quality. We all had a different idea of when that image became good to answer that question. So in this talk, I'm going to show you quite a bit of image processing, and none of it will be as good as the bit I'm about to show you now. So, strap in, everyone. So that first image is that image. I asked you if it was a good image. Some of you said yes, some of you said no. What I'm going to do to make that image good is I'm going to change the question. I'm going to say, when could you accurately count the number of nuclei? So here comes this image. Can we count the number of nuclei? Yeah, there we go. Thank you, everyone. That is image processing for you. So this is a really interesting point about fluorescence microscopy, right? Our image quality doesn't just depend on what the image looks like. It depends on what question we're asking and what information we want to get out of that image. So let's try and quantify this a bit more and see if there's a formal relationship between image quality and downstream analysis. So this data it's just a 2D plane from a 3D spheroid where the nuclei are same first. On the left-hand side, the raw data, just the same field of view where I'm turning up the laser power and acquiring. Next to it, you've got the result of a star disk segmentation. So literally getting an algorithm to say, where are the nuclei? What do they look like? And as you can see, the segmentation kind of changes depending on our illumination conditions. And our target is just the segmentation that I've done from a very high quality image. So that's what I want my segmentation result to look like. So what we can do is we can actually plot this out. We make this graph here on the y-axis. We've got the illumination dose that we're applying to the sample. But the x-axis, I do know my x and y-axes. I did do a high school maths. And on the y-axis, we've got this quantity called the AJI, the aggregated Jacquard index, which is basically just the measure of how accurate the segmentation is. So you can see we've got kind of three regimes maybe to this plot. We've got our kind of low illumination dose region, where small increases in illumination dose have a big impact on our analysis accuracy. We've got this region at the end, which is a plateau, where no matter how much more laser we put in, we're not getting more accurate in our analysis. And then we've got this little transitional shoulder in between, where you're kind of on the cusp of being okay. How about a different problem? So again, here's a different sample, a different analysis question. This time it's phalloidin actin in mammalian cells. Again, left-hand side, I'm just increasing the illumination dose. And next to it is the result of a type of analysis called AFT. And this is what I briefly explained earlier. Splits the images into blocks and says, what direction is the stuff in this block pointing? And again, our target image is the result of doing AFT analysis on a really good image of that field of view. But we can do a similar thing. So we can plot our illumination dose and our AFT accuracy against each other. And we get the same kind of shaped graph, right? We get a fast improvement, a slowing, and then a plateau. So if we come back to this question I asked us to begin with, what's the worst image I can still analyze accurately? Then it's probably somewhere around here. You want to be just past that shoulder. There's no point in imaging much lower than that because you're not going to get as good at information as you could. There's not much point imaging with higher illumination doses because then you'll bleach your sample, you'll kill your sample. You want to be just before that plateau. Okay, so that's how we know if images are good or not. Difficult to do, but that's our basic framework. It's a kind of this general relationship. Now, the problem is, as microscopists, we're never quite happy with what you've got. And we're always asking, how can we make images better? So there are a couple of ways that we can do this. So number one is we can improve the resolution of our images via super resolution microscopy. So this, for example, is where you have your diffraction limited image, you apply a super resolution technique, and you can suddenly improve your resolution by a factor of 2, 5, if you're really lucky, 10. And in a previous life, as Anna alluded to, I used to develop methods to do this. So this is an algorithm that I developed during my postdoc called SURF, which tries to extract super resolution information from just wide field fluorescence data. So it's purely image processing. Another way we can try and make our images better is via AI image restoration. And this is for example, a task you might do with this is you might have some very noisy data, and you might train a network to convert that noisy data into clean, high signal to noise data like this. So that's one example of AI image restoration. That's the CARE paper from Martin Weiger about 7 years ago. And AI image restoration is also, for better or worse, coming to a microscope near you. So this is some noisy data that I acquired, and there's a magic button on the Nikon microscope called denoise.ai, and it denoise.ai your data. So we can make our images better. We can improve the resolution, we can improve the noise characteristics. But how do we know if these images are better? So briefly, in super resolution, there's a lot of things that can go wrong. That's a happy note there. There are three main flavors of super resolution microscopy. There's STED, SIM, and SMLM. Super resolution never met an acronym it didn't want. So in STED microscopy, if this is our confocal image, instead of scanning with a confocal beam, we scan with this beam pair that restricts the emission to just the center of this little kind of donut shape. And so you get your nice super resolved image. However, everything comes at a cost. And here, the cost of resolution instead, one of the big costs is the cost of photons. So you're throwing away an awful lot of photons instead. Your images can be very noisy. Structured illumination microscopy, again, this is a wide field technique. So you'll have your stained sample. And instead of acquiring one image, you'll acquire a series of images that are illuminated with a different pattern. You then take these different patterned images, do some magical maths, which I don't think anyone quite really understands, but involves Fourier space. And you can reconstruct your image with a factor of 2-fold improvement in resolution. Now, again, of course, things can go wrong here. So again, anything that involves an algorithm is always trouble. So in SIM, if you get your reconstruction parameters wrong, your image will look weird. If your sample moves or bleaches between these different illumination patterns, your image will look weird. And so you can get these really funky kind of artifacts. This is a real classic SIM artifact of honeycomb pattern over your image. Finally, single molecule localization microscopy. This is again a wide field technique where instead of labeling our sample with a constantly emitting fluorophore, we label it with a photo-switchable, photoactivatable fluorophore where you can modulate the fluorescence on and off. And then computationally, you find the center point of each and every one of those molecules. And if you plot them out, you get a lovely improvement in resolution. And this is from the wonderful single molecule localization microscopy benchmarking challenge dataset, everyone's favorite image to reconstruct. But again, there's a computer involved, there are problems. So you must be able to identify individual molecules and find their centers very accurately. If you don't, the kind of artifact that you can expect, so these are microtubules, here's a zoom in. On the left, we've got microtubules that have collapsed onto each other because you haven't got the resolving power to see them separately. And on the right, we've got microtubules that look very fuzzy and you can't resolve them very sharply. So again, what's this question? How do we know if an image is better? How do you know if an image is good? So in super resolution, there are some techniques to do this. So for SIM, for example, there's a technique called SIM check, which can have a look at your raw data, check it for this bleaching, can check, make sure you've got the right reconstruction parameters, and it can check your final image for any artifacts. For For single molecule localization microscopy, there's a lovely technique called Hawkman from Susan Cox's group. And this looks out for that artificial sharpening artifact. So this will tell you at what resolution you can trust your data. So this tells you when your data's done that thing of you've got two objects, but the algorithm has smushed them together to make one. But what about more generally? Is there a general method we can use to measure quality in super resolution? So for many years, the way that we did this is you go to your paper, you publish your super resolution image, and to show that it was good, you just stick the wide field image next to it. And this is not the most stupid thing in the world to do. You can see it's basically kind of the same thing there. It's not the same resolution scales, but it's at least proving that this reconstructed image didn't come from nowhere. It's not total magic. So again, a long time ago now, one of the things I did in my postdoc was to develop an algorithm I told you that super resolution loves an acronym. I am no exception. So the algorithm is called squirrel. And that stands for super resolution quantitative image rating and reporting of error locations. And you can tell why I refer to it just as squirrel now. So what squirrel does is it takes our two images that you might put in your paper, your super resolution image and your diffraction limited image of the same structure. And essentially all it does is it formalizes what your eyes are doing. It compares the two. So what it does is it says that, right, let's imagine your super resolution image is perfect. If it's perfect, and I pass it through a virtual microscope, which is nothing more complicated than blurring it and scaling the intensity, it should perfectly match my wide field image if there are no artifacts. So you get your wide field image if your single molecule localization image was perfect. Now, spoiler alert, it probably isn't perfect. And the way that we can assay that is literally by comparing it with our wide field image. So we can create an error map. So the yellow areas are kind of danger zones where there's a mismatch between our blurred single molecule image and our wide field image. And we can also get a quality score. So this is just the resolution scale Pearson's correlation coefficient between our true wide field and our simulated wide field. So what kind of task could we use Squirrel for? So this is a lovely sample that we used in the Squirrel paper. And it turns out, and this data is from Christophe Leterrier, who is incredible at imaging all this kind of stuff. So it turns out that one of the big discoveries that super resolution has actually made is that actin along neurons actually forms little rings that are periodically spaced. And conveniently for super resolution, the periodicity of those rings is about 190 nanometers separated. So if we image these, again, fixed rat hippocampal neurons, where we stain the actin with LEX647, if we image in wide field, you don't see this striation, you don't see these rings appearing. It's beyond the resolution limit of our microscope. So what you might want to do is super resolve them. So you get your raw data, you get all these molecules blinking on and off, and then we're going to want to localize them to make an image. So the question is, how many frames of raw data do we need to make a good image? So you might guess a fairly large number, 500. 500 seems like a lot of images. If you use just 500 frames, then our image looks terrible, right? It's very undersampled. You can barely see the macro structure, let alone any nanostructure. So how much more do we need? So typically, the way that we calculate this in super resolution is you've got the exposure time to your camera, and you can calculate the amount of time that it takes for you to leave the microscope, get a nice cup of coffee, maybe a biscuit, drink the cup of coffee, eat the biscuit, come back, divide that number by your exposure time, there's your number of frames, the cup of tea method. And then you round it to a nice round number, and you get something like 60,000, and lo and behold, you can resolve your super resolution actin rings. But sad news for the fans of the coffee method, this isn't maybe the most scientific way we could go about this. So we decided to see if we could optimize this process with Squirrel. So what you're going to see in this movie is, in the big picture, I'm just going to add 500 frames more localizations each time. So I'm going to add more data to the image. In the top graph, we're going to plot out the quality metric from Squirrel, this resolution scaled Pearson's correlation coefficient. Higher is better. And in the bottom graph, we're just going to plot the resolution of the image. So there, lower means higher resolution, because resolution is a silly backward term. So let's see what happens. So again, it's our favorite relationship again, right? You add data, you get this fast improvement in resolution, fast improvement in quality, and then you hit a plateau, and you don't get any better. And then if we watch right at the end, we start actually getting worse, right? We add more data and our image gets worse. That shouldn't happen. So what we kind of thought was, right, when should we have stopped this acquisition? When did we get the best quality? When did we get the best resolution? And very frustratingly, it was not at exactly half the data. It was at 29,500 frames of my 60,000 frame acquisition. But that's when our quality value peaked, and that's when our resolution value reached its best value. And if you zoom in, that's what our reconstruction looks like from 29,500 frames. And this is what it looks like from 60,000 frames. So there's no more information that we're gaining by adding all that data. All we've done is we've wasted time at the microscope, we've wasted data storage, and we've actually made the image a bit worse. So it's not that we just wasted time. Because of that little dip at the end, over half the data required actually disrupted our experiment. And that's a nice open goal question at the end, if anyone wants to ask it, as to why that happens. So that's kind of nice. But again, I've been talking about the information in images. And one important question is, does what these quality metric reports actually reflect the information in the image, the thing we want to measure, the spacing of these actin rings? So we tested this in a couple of different ways. Firstly, there's a way that you can, for this kind of regular pattern, there's a metric called the visibility, which is a little bit of maths, which tells you how visible adjacent things are by looking at the peaks and the troughs between them. Happily, this visibility metric also peaked at the same point as our quality. We also did some frequency analysis. Again, we've got a lovely periodic structure, so if we do some Fourier domain analysis, we can see when we got the known spacing in these rings. And again, at the number of frames where we thought it was optimum, we had the strongest peak in the frequency analysis. So this is really reassuring. Maximum image quality, as measured by the squirrel metric, coincided with the maximum biological information content, the thing we wanted to get out of the image. So squirrel will tell you this kind of thing. It'll tell you how you're doing in your imaging. It's got a lot of limitations, but in general, it's a fairly decent method for seeing if you're in the right ballpark. Okay, so here's some data from that surf paper that I showed you earlier. This is a T cell that's been dropped onto a antibody-coated cover slip, actin is labeled. We've got turf imaging on the left, where you can see stuff is happening, and the surf process data on the right, where you can see the same data, but processed. There's a lot more contrast, there's more resolution. One of the things we wanted to quantify in this data was what direction the actin is flowing in. So what's happening? Is there a direction to where the actin is going? And we quantified this using a technique called particle image velocimetry. And if we look at the analysis of these two movies side by side, what we're expecting to see is a color map showing us what direction things are moving in. Applying the analysis to the turf image just doesn't really get us anything. You get a sad black hole. Whereas applying the same analysis to our surf data, I think it was meant to stop, you kind of recapitulate this color wheel, so it shows that everything's flowing to the center. So that's really good. This is an example of where super resolution microscopy could increase the biological information in our images. We saw that with the actin rings. We saw information that we couldn't measure from our diffraction limited data. And Squirrel will tell you if that data is good or not. But here's another image from the same surf paper that we published. And it's just demonstrating how good the algorithm is. I've got some microtubules in wide field. I've got the microtubules after being processed with surf. Now, what information have we actually gained by doing this processing? If the question is where are the microtubules, you could already get that from the wide field image. You did not need to super resolve it to tell you that. So while Squirrel will tell you about quality, it won't tell you if you needed to use the super resolution method to begin with. And that's just a cautionary tale of just because fancy methods exist, that doesn't mean they will help you. That doesn't mean it will answer your question any better. And there are artifacts and it can cause you more pain. So back to our how do we know if images are better after processing? For super resolution microscopy, we've got all these fantastic tools. We've got things like SimCheck and Hawkman, we've got Squirrel, and we've got wonderful benchmarking data sets hosted by BIOP that let you test out algorithms on data sets with known answers. And again, some research that I'm doing in collaboration with Susan Cox is actually trying to combine some of the single molecule techniques into a single software package, so you don't have to download 8 different things to see if your data is good. Okay, so what about AI image restoration? This feels like it could be the frontier. This feels like it could be useful. How do we know if images are better after we process them with AI? I have bad news. This is kind of the answer here. We don't. We have no idea if AI processing helps or not. So let's think about why we might want to do this in the first place. So let's revisit this plot. This was our plot of how segmentation accuracy evolved with our illumination dose on a sample. And we said that theoretically, the best image that we acquired would be there. That's the best compromise between illumination dose and analysis quality downstream. But just because that's theoretically the best image, that doesn't mean that's what your sample will tolerate. So it might be that practically all your sample will tolerate is down in that bad bit, right? Down where your analysis quality isn't good enough. That might be where your sample dies, where your sample bleaches. That might be actually the best you can do on the day. So how can we rescue this kind of image that's not good enough? Well, our answer, image processing. So we've probably all done the most simple kind of image processing there is, which is literally just a local mean filter. Just locally average your data, and you'll see that visually, actually, it looks pretty good. But where AI is coming in is trying to do this a bit better. So you can take your not so good data, and you could run it through a pre-trained AI. So this is the kind of thing that's a button on your microscope. And you can see, maybe looking a bit better than our local mean filter. Or if you're feeling particularly extra and just like fiddling around in Python, you can train one of the available open source methods, and you can train an AI that will denoise your data specifically for the kind of images you're looking at, and you'll get something that looks like this. So I told you we don't know if they help or not, but we kind of do, right? We know if they'll help if you know what the answer is. So we can know if they help. So obviously, when you're developing an image processing algorithm, you need to benchmark it. You need to know if you've got better or worse. So this is how it's done in the literature. So let's say this is my noisy test image. We don't have any quality metrics for a single fluorescence microscopy image that'll tell us how good it is, right? We just don't have any. The only way we know how good that image is to compare it with a better version of itself, which is a totally stupid thing to do data at the microscope. If you could acquire the middle image one, don't acquire the one on the left, just acquire the good one. But let's say we wanted to measure the quality of an image. This is how we do it. We compare it to some kind of reference image. And the metrics that we normally use for this are things like normalized root mean square error, peak signal to noise ratio, the structural similarity index measure, and Pearson's correlation coefficient, a whole host more acronyms. And these ones in particular that are used very often in the literature, we've stolen from computer vision. Pearson's correlation, that's statistics, but these guys we've stolen from computer vision. So if we use those, we can say, right, this is the quality of our test image. What we'll then do is we'll then take our super duper image processing and process our test image, and it'll look like this. We can then compare that to what we know to be our reference and recalculate our metrics. So the idea is we're looking to compare the metric values before and after processing with this reference of what we want the image to look like as kind of gold standard. And just to show you, this is generally how it's done in the literature. Anytime you'll see some kind of AI image processing algorithm published, there'll be a graph like this. It'll be a graph that's showing several different methods, several different conditions, and how these values change depending on the processing that's applied. And of course, the one that scores best will be the one in the paper, because that's how we roll. Okay. So here's a question. Are there reasons to be suspicious of these metrics? Should we trust these quality metrics? Now, I told you that at least 3 of these are stolen directly from computer vision. And the kind of image that we're used to looking at in computer vision are natural scene images. And natural scene images are basically all this. They're all pictures of my cats. And they're beautiful and they're cute. And if you've been paying attention, you'll notice they don't look that much like fluorescence microscopy images. They look pretty different, namely lack of cat, but also there are some other differences that we'll go through. So why are these not the same? Firstly, the bit depth of the images. Natural scene images are almost always 8-bit or 8-bit RGB images. And if you have a look at the histograms for my lovely cats, you'll see that most of the grey values from 0 to 255 are represented in that image. The histograms go all the way up. In fluorescence microscopy, however, we have a lot of different bit depths. We could have 8-bit, 12-bit, 16-bit data. And we almost always underfill that bit depth. So these are 16-bit images, and the maximum values that pixels could take in these are 65,335. If we look at the histograms, we are nowhere near that, right? We're all squished down right at the bottom. We also have differences in the noise. So again, in our fluorescence microscopy images, you can really see noise. Whereas in natural scene images, you don't really tend to see noise. You only see noise or artifacts if you've done something weird like compressed the image. And finally, sparsity. In my natural scene images, every single pixel in that image provides a lot of information, mainly about how cute my cats are. Whereas in fluorescence microscopy, look at all those black pixels. Sometimes 90% of the pixels in your image will just contain no information. So it's very sparse data. So this kind of slightly bothered me. So what I decided to do was actually see, do these metrics reflect image quality in microscopy? So what do we expect them to do? What do we want them to do? So we expect that if we physically improve our image quality by shining more light on our sample, then these metric values should go up. That's like the bare minimum they should do. So again, here's one of my little tests. So the reference image on the right is what I'm comparing to. And I've got my series of test images just at different illumination doses. So let's see how the metrics behave. Pearson's correlation coefficient, good. It goes up. That's good news. Peak signal to noise ratio, again, we increase our illumination dose. The metric value goes up. Structured similarity index also does the same thing. So this is great news. This is fantastic. Expectation matches reality. We increase our image quality, and these metrics say, yeah, your image got better. So that's good news. We're off to a good start. All right, what's another expectation we might have? So leading on from this, we might expect that different images with the same quality metric value have the same quality, right? That feels obvious. So let's look at some pairs of images. So these are all crops from a bigger image, but it's just to show you the detail. So these images have the same Pearson's correlation coefficient, and they don't have the same quality visually. These crops are from two images that have the same peak signal to noise ratio. They don't have the same quality. And in an absolute shocker, these are two crops from two images that have the same structured similarity index, and they don't have the same quality. So our expectation is not being matched here. And why is it not being matched? So remember I said sparsity is a big difference between fluorescence microscopy data and natural scene images. So these are where I take, these are the whole images from our SSIM crops. And you can see one is much sparser than the other. So in this case, even though it was kind of worse, this SSIM metric scored this denser image as better than our sparse image. And this is something that we've seen really repeatedly, and you can actually measure it and model it. It's a real phenomenon. So our reality here is that image sparsity can bias our metric values, which is kind of tricky. We don't want it to do that. We just want our metric values to report quality, that we don't want to report sparsity as well. All right, let's have another expectation. So if you've got some different structures, if those images have the same kind of quality metric value, then they should be of the same quality, right? So again, I've got a load of different, so I've got some images here, 4 different structures, and then we've synthetically degraded them. So we've added the same amount of noise to each one. And we can calculate quality metrics for different types of structure. And what you see is different quality metrics just favor different kinds of structures. So here, the Pearson's correlation coefficient really favors our DNA images. They will always score higher than, for example, our fibrillary images. Peak signal to noise ratio kind of switches around. Now our DNA not so hot, COX-4 though. Pearson R always really likes the COX-4 marker. And again, for the S, it's another order of preference. So again, image content can play a really significant role, more so than the amount of noise in the image. What you are looking at seems to be as important, or if not more important, than the actual signal to noise of that image. But remember, no one uses it for this, right? We're not looking at raw data. We don't use these image metrics to just assess raw data. We use them in the context of before and after. We use them for image processing. And so are they meaningful after image processing? So this is the kind of summary figure that you'd see if you looked in a paper that was comparing different image processing methods. We assess them using these different metrics. And again, our expectations, higher metric values mean higher quality. That is what we want these things to us, and that's the assumption. So let's see if that's the case. So again, this is just a crop from a bigger image, but this is the reference image that I'll be using. It's just yeast filled with some GFP. You can see some inclusions in the cells. So for this experiment, again, for the same images, I've acquired a series of raw, noisy images and then denoised them. And what we're going to do is see how well the denoised data performs. So just mean filtering one of these noisy images will give you this. It kind of does okay, but you lose some of the fine structure. For example, the kind of tubular bit between those two pink arrows, that's gone. If I denoise one of my raw images using the pre-trained AI, we do a bit of a better job, right? We can see, oh, we've maintained some structure here, so we've got some structure back. That seems pretty good. And if I denoise one of these noisy images using a custom AI, you'll see this is, with the best one in the world, complete garbage. That is not what it is. So we've got 3 processed images with 3 different qualities. You'd expect them to have different quality metric values, but if we compare each of these images to our reference image, they all have the same peak signal to noise ratio, which is insane. And this is kind of seems to be a real feature of these quality metrics. Any kind of image processing seems to improve the quality metric values, even if the images themselves end up looking worse. So this leads us on to kind of 2 big questions. Firstly, and I've made them actually quite big. Are image quality metrics ever useful? Should we trust these things at all? Can they ever be helpful? And secondly, more controversially, is denoising ever useful? Is this AI processing that I've shown you, can that ever help you out? So what I've been saying all along is our measure of quality needs to be how well we can answer a biological question downstream. So let's have a look at a few examples of that and see how denoising and our metrics perform. So firstly, let's look at our old friend segmentation accuracy. Can quality metrics predict segmentation accuracy? And does denoising help segmentation? So this is our experiment. Again, it's Hurst staining nuclei in fixed spheroids. And I've got, this is going to be my reference image, and this is my reference analysis. So these are the things I'm going to be comparing my image quality metrics and my analysis accuracy against. So here's my raw data. Again, different illumination doses. And I've calculated here, just in this example, the SSIM value for the raw data and the analysis accuracy for the analyzed images. Then what we've also done is we've applied a load of different denoising methods, that raw data, recalculated the quality metric, and redone the segmentation. So we can see if the segmentation gets better if you do it to the denoised data. So I'm going to show you a plot. Ignore the data points to begin with, because I'm just going to talk about the axes, because it's a slightly complicated plot, I'm afraid. So what is going on in this plot? So on the x-axis, what we're saying is this is the change in quality metric value, right? This is the thing that we're plotting in our image processing papers, where we assume that a large improvement means your image got better. So a large improvement in the x-axis means that the quality metric thinks that denoising made your image better. On the y-axis, we've got the change in segmentation accuracy. So again, a higher value means your segmentation was better after denoising. A lower value means a negative value would mean your segmentation got worse after denoising. So we've got these 4 quadrants. We've got sensible quadrant 1. The image quality metric said you got better and your analysis got better. And we've got sensible quadrant 2. Your image quality metric said the image was worse and your analysis got worse. Then we've got silly quadrant. We've got silly quadrant where the metric says it got worse, but the analysis got better. And we've got this really silly and quite dangerous quadrant where your image quality metric was like, yeah, you did great, but the analysis got worse. So the data points we have on here are, there's some aggregated data for mean filtered images. So it's the same story seen before. Even though mean filtering is not, you can't get more information by mean filtering, really. If anything, you're reducing the amount of information. But you can see the quality metric goes up quite a lot just by mean filtering your data. But all of our analysis data points, they're on 0. You haven't improved your segmentation at all. Even though the metric was like, woo, we got so much better, you've got no improvement in your analysis. My pre-trained AI, this is kind of sad times. So again, we're almost always improving our quality metric value. But here, we're also almost always making our segmentation less accurate, especially for that little guy down there. that was a particular set of illuminations where it really likes, the denoising thought it was doing a great job, but the analysis just, I couldn't get it to work at all. And then what about a custom trained AI? Well, here it's slightly better news, I guess. I mean, again, our quality metric values have gone up, and we get a very slight bit of correlation with a very small improvement in segmentation accuracy, but not going to set the world on fire, like a 4% improvement. So our denoising doesn't necessarily help for this problem. For this particular data set, for this particular problem, the denoising doesn't necessarily help. And our improvement in metric scores doesn't necessarily mean that we've improved our actual quality, our actual biological information. All right, so that's bad news for metrics, bad news for denoising. Let's move on to problem 2. So this is our filament analysis accuracy. So again, just as an example, here's going to be my reference image that I'm comparing to, and here's my reference analysis. Again, we've got images at different illumination intensities where we calculate our metrics and our analysis accuracy. And then the same thing after denoising, okay? So let's see where this sits on our little plot. So we mean filter the images. What happens? Again, we improve our quality metric scores. Sometimes the image gets better, sometimes the analysis gets worse. Our metrics don't really tell us anything here. It's just this kind of cloud of confusion. So that's not a great start for our metrics. Our pre-trained AI is a little more useful, right? In this case, our denoising, apart from these kind of left-handmost points, which is where the image was very good to begin with and you didn't need to denoise it, actually, you almost always help your analysis accuracy. For this problem, denoising will help you. Quality metrics kind of tell you that, kind of don't. But then for our custom trained AI, again, this is good news for everything. If we process our data with a custom trained AI before doing our analysis gets better, and good news, the metric says we get better as well. So it's not all bad news for our metrics. The right combination of task and denoising can help your analysis, and sometimes can be supported by the metrics that we use to assess this. Let's have one final little task. So classification accuracy. So what is in the image? Can you tell what's in the image before and after denoising? So this is done in collaboration with Andrew Gunawan down at University of New South Wales. And what we did was you've got loads of images from the Human Protein Atlas classification channel, so images of different subcellular structures. And then we generated synthetic noisy images from this data. And then with different methods, we denoised them. For our noisy and our denoised images, we then trained classifiers to say, can you tell me what's in this image? We know what it should be. Can you learn from the noisy images what's in there? Can you learn from the denoised images what's in there? So what we want to happen is something like this. If our reference image is this, it's an image of the plasma membrane. In your noisy data, it might wrongly classify it. It might think it's the cytosol. And after denoising, it might get it right and say it's plasma membrane. These are all real examples. However, what we also saw was this example. Our reference image we know is endoplasmic reticulum. Once you add some noise, it gets confused. It thinks it's plasma membrane. But the denoising still doesn't help. It denoises it, but now it's classified as the cytosol. So that's not helpful. And our final example, nuclear membrane, again, here in this case, the noisy data is like, oh, yeah, I know him. That's the nuclear membrane. Whereas after denoising, it gets it wrong. So again, this is another problem where it doesn't really seem like we just don't know if this will help or not. And do the quality metrics tell us anything? Not really. All these pairs of images have a change in PSNR of 4. Each of our denoised images is 4 PSNRs better than the raw image. So it's really unclear. So the usefulness of, just to really underline this, the usefulness of denoising and metrics really depends on both the sample that you're looking at and the task you're performing. Finally, to finish off, the question that's usually end every talk with, which is what's the worst that can happen? So this is some 3-color imaging I've done. And the reference image that I'm going to use here is mitochondria. So I've got a nice high quality image of some mitochondria here. That looks pretty nice. What I've been doing the whole time is imaging the same structure, but at a lower illumination dose. And so you can see it's the same structure. You can tell it's the same structure, but it's kind of noisy. And if we put that through our quality metrics, they are underwhelmed, to say the least. They're like, not a great image. Okay, let's do something silly. Let's change color and let's image these same cells in the green channel, which here is actin. So I've got a nice high quality actin image, but it's clearly not the same structure. Let's see how my quality metrics rate my actin compared to my reference mitochondria. Okay, so we've increased our PSNR value and we've increased our SSIM value, but our Pearson's correlation hasn't got any better. And you might think, oh, well, that's you know, being a bit facetious here. They're kind of the same shape. They're kind of the same intensity distribution. It's okay for metrics to get it wrong every so often. All right, let's look at the third channel, which is the nuclei. Again, we've got quite a different intensity range to our reference image, and we've got almost the opposite in terms of structure. How do the quality metrics rank our nuclei image against our reference mitochondria? Again, we've actually got better PSNR and SSIM values than for the real structure. And so our three different metrics tell us that the three different images are the best match to our reference image. And this is not useful for biology. In biology, structure is as important, if not more, than intensity. We really care about structure. And metrics just don't know anything about biology. So to conclude, what have we learned today? Firstly, the quality metrics that we use for benchmarking image processing in microscopy were not designed for microscopy. They were designed for a different task altogether. And we shouldn't really be surprised that they behave weirdly. They also have this tendency to just like processed images. So no matter what you do to your image, they tend to score them quite highly. And finally, image processing doesn't necessarily lead to improved downstream analysis. Sometimes it will. Sometimes you can really get good gains from processing your data before you analyze it. But there's no real way to tell if they will or not. These image quality metrics won't tell you. The only way to do it is to try. And because this is kind of a bummer to end on, we'll end on some more cat content. So this is Artemis, my cat. She's a visual representation of how image quality metrics work. There she goes. So close, yet so far. Thank you so much for listening. I'd really like to acknowledge our collaborators down under in Australia, especially Andrew, who's done all the classification work. Leads to interesting time zone Zoom calls, but it's worth it. Nandini, who did all the spheroid preparation that she kindly let me steal. And Richard, who's done a whole lot of maths behind the scenes that actually tell us that the behavior we see these quality metrics having in fluorescence microscopy can be predicted from the equations. And thank you